Hi guys, welcome back to Bye Buddy, and sorry for the late video, but I was stuck somewhere in some work. I'm really sorry for it. So I thought let's just continue with our work, with our videos. So today I've brought you with the Cdna synthesis, and the Cdna synthesis, as you all know, it's a very simple process, and I've made it more simple by this explanation. So if you want to know, just click to my channel, subscribe, like the video, and comment down below. So let's just start. What is Cdna? First of all, we need to know the full form of Cdna. Cdna stands for complementary DNA, right? So Cdna is a double-stranded DNA. Earlier it is not a double-stranded, I think it is made from the mRNA template, right? So Cdna is a complementary DNA which is made by the mRNA template using the enzyme reverse transcriptase. One side is methylated, on the other side we are adding poly A tail to the 3' end of that mRNA strand, right? So this mRNA strand is being used as a template strand, and you know what is the meaning of template, I hope. So template strand is used at the starter point to begin the synthesis of the cDNA. In this video, we will be studying all these steps which help us in synthesizing the cDNA from an mRNA template strand. So let's get started, guys. So this is a methylated cap, and on this 3' end we are adding poly A tail. This AAA plus A N means there are many A's which are present on this 3' end. To this, now we are adding oligo DT with the process of hybridization, right? With the help of that, we are adding here oligo DT. Let me just write it down. Oligo DT is added here, right? So now what we are doing, this reverse transcriptase, it is an RNA dependent DNA polymerase, right? It is totally depending upon an RNA. We are taking an mRNA, right? So mRNA is also a type of RNA strand. So reverse transcriptase is a RNA dependent DNA polymerase. So this is helping in our synthesis process. So reverse transcriptase along with the DNTPs, that is the 4 parts, adenine, guanine, thymine and cytosine, all these together along with pH 7 help in the process of synthesis of cDNA, right? So this reverse transcriptase is taken from a retrovirus, can be MMLV or can be AMV, right? So these viruses, now they are obtained, all this process is done. Now what we are doing to the poly A tail, this starts synthesizing in the opposite direction of that of the mRNA strand. This is our cDNA strand which is being synthesized, right? So now what is happening after this synthesis, the synthesis has begun. Now what we are doing, I already told you, we have hybridized oligo DT to the 3' end, that is T12 to T18 in number, in base pairs. Number of base pairs will be T12 to T18. Now what is happening, this totally has been synthesized, and now it is formed the mRNA cDNA hybrid. That means the mixture of the two different strands are present here, that is upon the mRNA template what has been synthesized. A new strand has been synthesized which is now being referred to as the cDNA strand. So this whole structure will be called as mRNA cDNA hybrid, because in this both the strands are present, one of the mRNA, other of the cDNA. Now what is happening, let me just come this side. Now the denaturation process happens, that is, as soon as this hybrid has been formed, we need to separate these two things to avoid any kind of infection or any kind of bacterial disorders which can spoil all this, right? Any kind of bacterial infection which can spoil. So what we do, we boil it at high temperature and we add RNase H that help in separation, right? That helps in the detachment. Now what we are doing after the denaturation of the cDNA mRNA hybrid, we know we have to separate it. We are adding two things, RNase H, and we are boiling it at high temperature. Now after the boiling has been done, it has been separated from the mRNA. So what is left now? Only the cDNA. cDNA is left, but always we know that DNA is a double strand, right? So this will be a single stranded cDNA because of the single strand which is present, and it is having T12 to T18 oligo DT primers which were added earlier, right? Now what happens? The synthesis begins, and the synthesis begins in the form of hairpin loop. Hairpin loop means a loop-like structure is being formed on the side. So you can see the synthesis is being started 3' and this is 5', and this helps in the intermolecular base pairing. With the help of intermolecular base pairing, only the synthesis is possible, because when they will pair up, then only the bases will be forming a intermolecular lock-like structure which helps in the formation of this loop. Now this loop has to extend further in order to form a complete strand. So what we are doing, we are adding another DNA polymerase, which is only DNA polymerase, and along with that we are adding Mg2+ ions that helps in the extension of the upper segment, right? Now the upper segment has been formed completely. These two segments are being formed, but they are connected through this loop-like structure which was helping in the extension earlier, right? So now what we have to do, we have to separate these two. How do we separate it? We are adding S1 nuclease. S1 nuclease means it helps in the separation of the two double-stranded DNA, which is first locked because of this presence of hairpin loop, but this hairpin loop has to be removed, has to be detached. So what we are adding to it, we are adding S1 nuclease, which helps in the detachment of both of them and makes them single. So this will be known as cDNA, which is now a double-stranded cDNA. So this process begins with a template strand, right? So this template strand helps in the further process of synthesis of the cDNA. Earlier a small cDNA started, then it enlarges, we have to detach it, we detach it. Now from the detachment it starts making another loop-like structure. Again it helps in the process of separation with the help of S1 nuclease, and after that, you know what happens, we get a double-stranded cDNA, right? So what is the main role? Who is playing the main role here? The main role is being played by this reverse transcriptase, because this is RNA dependent DNA polymerase, right? So this only is helping in the process of formation of the cDNA synthesis. I hope you guys like this video, because it was the easiest way possible. You could have understood this, I hope. And in the next video I'll be showing you how to form a genomic library, and how to form a genomic, what is the main difference between the cDNA library, cDNA, and the genomic library. So stay tuned for the next videos, guys, and won't be late this time. So please subscribe to my channel, like the videos, and comment down below, and if any doubts, do ask below. Thank you, guys.